In this question, as you can see, this is the structure of human eye. Now, the question is, when light rays enters into eye, the most of the diffraction occurs at what? So the most of the diffraction you should know that takes place from the outer portion of the eye, right? That's called cornea. And what is cornea here? This is the outer bulge portion of this eye. This is called cornea. And I can see this is marked F over here, right? So F is the correct answer. Now we should also know why. Therefore, the question arises then what is the use of this lens? Well, the lens is, you can see this is convex lens. The question is, what is the use of this convex lens if the refraction takes place? Because ordinarily we know that the lens actually should do the role of refraction, but that's not true in case of eyes. Most of the refraction takes place to the outer portion of the eye. Now, what is the role of this lens? You see here, this lens contract, sorry, it thickens and become thin so as to keep on adjusting the focal length of the eye so as the image is always formed in the retina. I should repeat, as you can see that it is getting thin when you are looking into the far of things, focal length increases, become thick when you are seeing nearby objects, and this is done so that the image is always formed in the retina. Unlike camera, we do not have different lenses fitted, so we just have one lens. The thickening and thinning of these lenses helps in forming the image at the screen of the eye called the retina. I've taken two questions here. The first one, I'll do this one. It says select the figure that represent the correct magnetic field lines. There are two magnetic field lines as you can see here, and we also have to tell the reason. Now, first of all, you can see that what you know, no two magnetic field lines are found to intersect each other. Well, the reason is very clear that if they were to intersect, then at the point of intersection, if you place a magnetic compass, then the needle will point towards two different directions, right? Which is practically not possible. That's the reason we will give a justification that this is an incorrect diagram, as two magnetic field lines are never found to intersect each other. Look into this. The diagram is correct, this one. But then we also have to name the poles facing each other. What you know that magnetic field lines arise from the north and merge at the south. So this must be the north and this other end has to be south. Here too you can see the magnetic field lines are arising from this end. So this is going to be north and this is south, right? That was the first question. Now coming to the next part of this, what does the second question says? State the rule that determines the magnetic field, direction of magnetic field produced in a straight current carrying conductor. So that's what that is right hand thumb rule. And I think this is very clear to all students that if you hold a current carrying conductor with your right hand in such a way that the thumb points to the direction of the current, then the fingers wrapped around it indicates the direction of magnetic field. And this is what concentric circles, right? That was the first, and there is second part also. It says force experienced by a straight current carrying conductor placed in a magnetic field. So here the answer for this will be what? Let me erase the later part so you're getting confused. Now here we are talking about the Fleming's left hand rule. It is the Fleming's left hand rule which determines the direction of force produced in a conductor placed in a current carrying conductor placed in a magnetic field. So they are represented as this. So if you if you stretch your forefinger, the middle finger and thumb so that they are mutually perpendicular to each other. Here I can show you that these three are mutually perpendicular to each other. So if your the forefinger points to the direction of the current, you can take this as the forefinger, and the middle finger points to the direction of the current, then the force is represented by the thumb. So the force in which the direction in which the force is acting is indicated by the direction in which the thumb is pointing. Some questions have been asked from this section of your textbook chapter environment. It says cooler, which is a disposable cups, right, is made up of clay and disposable paper cup. Both are used as an alternative for disposable cups. Now our question is, which one can be considered as a better alternative cups and why? Well, most of you end up writing this cooler made up of clay as it is it will does not lead to deforestation. But when given an option, we should go for out of these two, right? Out of the paper, right? Out of the paper cups and the cooler, one should choose the option disposable paper cup. As you know that coolers that is disposable again is made up of clay, but this is not a better alternative. As you know that the if making coolers in large scale would result in loss of top fertile soil, right? Moreover, there is a logistic problem that is producing large amount of this much requirement of railways for disposable cup could not be met by any farmer or any. So these coolers cannot be produced in factories in large scale. That's why now disposable paper cup is used instead of plastic or cooler. So when given an option, we will choose for the disposable paper cup, and we should write down the reason or the reason for choosing these alternatives, because if we use cooler, then that results in number one, loss of top fertile soil, and number two reason over here will be that logistic problem, producing large amount of coolers as per requirement cannot be met by any farmers or by any other means. Then in the next question, it says name the reproductive and non-reproductive parts of bread mold. As you can see, I've taken one diagram from the textbook NCERT. So the reproductive part is what? This spongium is the reproductive part. These are spongium, right? These are the reproductive part, right? And this blob-like structure, right? This is blob-like structure, and the non-reproductive part is the thread-like structure. So this thread-like structures that is called hyphae. So these are called hyphae. I should write down. This is we call them as hyphae. This is HYPHAA, right? So this is the non-reproductive part of this rhizopus. Now the next question is, although from the different topic, not related to the diagram, is advantage of vegetative propagation. So what is vegetative propagation? Growing of a new plant from its vegetative parts, means other than reproductive part. Vegetative parts means what? The could be root, could be a leaf, or maybe stem, right? So the advantage of growing a plant through its vegetative parts is number one, that the plants produced are genetically similar. And the second advantage, that those plants, like in case of banana, banana has lost the seeds, lost the capacity to produce seeds, but we know we can continue to grow banana. That is because of vegetative propagation. The other advantage could be that those plants which are grown through vegetative propagation starts to bear fruits, flowers, right, much earlier than those which are grown through seeds. So these are the three important advantages. In this question, you can see this is the diagram of hydra, and the question here is, how does the bud develop into hydra? So let's figure out. What you should know, that a bulb-like projection, right, is formed over here, and it the cell inside, right, the bud develops at this site. There is a cell division takes place rapidly, and that gives rise to a structure, right? This is called bud, that grows and can remain attached to the parent or can get detached, right? In many cases, it get detached from the parent plant and grows it as a new organism. It get detached with this main parent plant, and you can see that it grows as a new individual, right? New individual. Now the question here is this, but then I can see the first option is only correct. The bud develops due to repeated cell division at the specific site. Well, the specific site, as I said, is this bud over here, right? Other options are incorrect because the bud develops due to separation of the body parts of the hydra is incorrect. Many a times it remain attached to the parent plant, but develops due to the attachment of another hydra at a specific site is incorrect. And bud develops due to change in the environment condition, obviously totally incorrect. So we'll say that this is the correct answer. In this question, it says an object when placed at the center of curvature, the image formed at the center of curvature. As of reading till here, we don't get what is it, whether it's a mirror or lens, because magnification as -1 is formed in a concave mirror as well as in case of a convex lens. Further, it says a student obtains a blurred image of the distant object on a screen using a convex lens. That's why I've taken a convex lens, and you can figure out when the object is at 2f, which is C only, the center of curvature, the image is formed at 2f2, that is other side of the lens, and it is real inverted and equal in size, and that's why its magnification is 1. To obtain a distinct image, well, in his case, the image is what? Here it is blurred. Maybe because he has not placed the screen at the right position. So in order to obtain a sharp image, he should move the screen depending upon the position of the object. It should move either towards the lens or away from the lens, right? That's what we get to know whenever we try to capture an image, and if you do not able to capture if you get a blurred image, then we change the relative position of the lens and the screen, right? And that's the reason we will say that the correct option will be the option C here. One more time, the correct option will be option C in this case. The question here is which of the following statements are true about the endocrine glands? So what we know that endocrine glands are what? These are ductless glands. That's true because they do not release, unlike liver and pancreas, they don't have duct. They directly released into the blood, right? And the blood takes it to its target site. Yeah, this is also given. They release their hormones directly into the bloodstream and it goes to the target site. Further, I can see that they say that they release their hormones into the duct. So this is incorrect, right? They don't release hormones into the duct. That's why they are called ductless gland. And then they produce chemical messengers, right? That's called hormones. So these protein are also called hormones. So we can see that the option 1, 3, and 4 is correct. 1, 3, and 4 is 1, 3, and 4 is this. So we'll say that the correct option here is option A. In this question from the chapter life process excretion part, it says a given figure represents a single nephron from a mammal kidney, that is a human kidney. In this case, we have to identify the label parts and match with the options given here. Now, what you can see, the first they are asking for the site of filtration. What you know that filtration or the ultrafiltration takes place at the glomerulus, right? So it takes place at the glomerulus. I can once again show you. This is what A is labeled. A is glomerulus, where this is Bowman's capsule, and inside the blood capillaries does the ultrafiltration. So this is A here. So I can mark this is A marked correctly. Now, if I check the next one here, the second one is collecting the urine and make it more concentrated. Has to be DCT, that is the collecting duct. And the duct here, I can see it is marked as E. Right, this is E here. Now, next is the main site of glucose reabsorption. So you know that the useful substances, not only glucose, the other substances, glucose, amino acids, and some salts are reabsorbed at this, that is called the tubular part of the nephron. And lastly, we are left with what is it? Next is responsible for maintenance of pH, and one which is responsible for maintenance of pH. Now we are left with what? D, that is this is. So therefore we can see that B is the correct option in this case. We'll go for B in this case. I've taken two questions from the chapter Reproduction class 10th. The question here is give reason the placenta is extremely essential for the fetus development. Now let's understand where is placenta. Placenta is actually the tissue. You can see this is the tissue which is present in the uterus, and that its main role is to provide nutrition to the growing fetus. Right? Nutrition here, as you know, that the baby should get the growing, not baby, we'll say the fetus should obtain nutrition, and this placenta provides the nutrition over here. And not only the nutrition, but also the nitrogenous waste produced by the fetus is also come back to the mother's body through this only. So as the urea, uric acid, and the other nitrogenous waste gets removed from the fetus body. Now the second one is uterine wall becomes thick. As you can see, there is a thick lining of uterus, and not now. The moment the egg is released, the uterus prepare itself to receive the zygote. The uterine wall becomes thick to receive the zygote. This is what. And if the fertilization does not take place, then this, along with the egg, is shed from the body in the form of blood, which we know as the uh, menstruation, right? So we are required to just answer, we just need to say that this is required for nourishing the embryo if fertilization takes place, and this we have already discussed. We have one question here to find the difference between a glass slab and a glass prism, and later part is what happens when a narrow beam of monochromatic light and a white light enters to a glass slab and glass prism. So let's just begin with the monochromatic light here. So what I'll do here, let's say if I take one incident ray here, striking the glass slab here. First I've taken a glass slab. As you know that in case of refraction, what will happen as the ray is traveling from a optically denser to rarer to denser, it should bend towards normal. One more time, if undeviated, it will go like this, but a deviation is taking place. An undeviated one will go like this. But as I told you that here the deviation should takes place. So this is what the dotted line indicating the produced part of incident ray. But the ray will deviate towards normal. So this is what it is bending towards normal. And if I make a normal over here also, the emergent ray will come out from here, right? Bending away from normal this time. This will bend away from normal. So the emergent ray will become parallel to the incident. Well, this is what this length is called lateral displacement. So what you can figure out that in case of a glass prism, this is not a prism, this is a glass slab. Here the incident ray is parallel to the refracted ray. Now check what happens in case of a monochromatic light if it enters into a glass prism. This is an incident ray over here, and this is a normal over here perpendicular to this lens. So it will refract here, it will bend towards normal, and the emergent ray will once again bend in this direction. Now, if I produce this emergent ray backward, right? If I produce this emergent ray backward in this direction and the incident ray forward, these are produced, as I said, these are produced rays. So it's going to be dotted over here, right? What you can figure out, there's these rays actually meet with some angle, right? And they meet, they make an angle. So this is the main difference between the monochromatic light when it strikes a glass slab and a glass prism. In case of a glass slab, they are parallel. In case of a glass prism, they meet, right? They intersect. Now, but when I do with the next part over here, the next part is to figure out that was for a monochromatic light. Now it's a white light. So as you know that in case of a white light, there will be splitting of white light into its component color. That's called dispersion. So the dispersion will take place where spectrum will be formed in case of a glass slab, as you know that VIBGYOR, right? So here that in case of a glass prism, there will be dispersion and no dispersion will takes place. No dispersion will takes place in case of a glass slab. Well, we are talking about the second part here for white light. For monochromatic light, that's the difference which I said.